I have the honor of being on stage with Dr. Ben Brown of the Lawrence Berkeley National Laboratory. And you might ask, why am I up here and not in the background? Is because I grew up on a farm in the 50s and 60s, and that's been about 60 years ago, where things were a little bit more calm and a little bit more easy, except the hard work and doing it by hand. But over the last 9 years, I've been working with the plant biome and soil biome and how fantastic it reacts to various inputs. And I think when Mark and Larry were on one of their annual trips, and Larry has this human biome that he's been following for most of his life. Probably how long, Larry? 15 years? But they started saying, why not agriculture? Where is agriculture in the FiRe conference? So when we started looking at that, there's a symbiotic relationship between the plant and soil biome and the human biome. And Dr. Brown, I'm going to call him Ben, one more thing. Kevin Montgomery tweeted out yesterday that this is the smartest place on the planet. I've got to agree with that. That applauds to all of you. Ben heads the M2B, or Microbes to Biome initiative at the lab, and I'm going to read what this is. Microbes to Biome is a Berkeley lab initiative designed to reveal, decode, and harness microbes, the most abundant and diverse life forms on Earth, in an effort to develop novel approaches for protecting fuel and food supplies, increasing environmental security, and promoting the health of the biological systems. To lessen the impact of our growing population, scientists are developing methods to produce more with less in order to reduce stress on climate, environmental quality, and health. Why is that important? I think we might be a little bit in trouble in what's going on in the air, in our soil, and our water system. So, Ben, it's an honor to be here with you. Can you give us sort of a 70,000-foot view of what you're seeing in the background of the biomes, Microbes to Biome?
Absolutely. Thanks so much, David. I really appreciate the invite. I appreciate being here. Thank you so much. So I'm going to tell you a little bit about our work in this area, microbiomes. The M2B consortium is very large, involving many folks at LBNL. I'm involved in a part of it. It's headed by Owen Brody, very talented colleague of mine. And the work goes all the way from metazoan microbiomes and health applications to agricultural. And the program really acknowledges the deep synergies and fundamental governing principles that are relevant to both animal and plant microbiomes in the way that microbes are used by both plant and animal systems. I myself, my background, I'm a statistical biologist. A lot of the work I do is actually in the area of ecotoxicology. All right, so I'm going to start to tie in how that's relevant. And since we've got a lot of human health folks here, I'm going to start out on the human side, and then we're going to take a quick walk into the ag side. Right. So seen behind me here, these are tap water samples from Maywood, California. Okay, when residents asked for cleaner water, they were told this meets the Safe Drinking Water Act. And that's a really interesting facet of toxic substances regulation in the US, which is we regulate against things we know are toxic. We don't necessarily have standards for unknown compounds. Those are developed in real time by the EPA as they come to market. So if you've got a bunch of unknown stuff we haven't seen before turning up in tap water, there's absolutely no law against it, and you're sort of stuck until the EPA figures out something to do. And this comes up all the time. Many of you remember the 4-methylcyclohexane methanol spill, the MCHM spill, into the Elk River in West Virginia a couple years back. There was 70,000 gallons of this compound used in coal refining and fracking into a drinking water source that 1 million people depend upon. There are absolutely no standards for this, no standards for what levels it could be at in water, no standards for lethal dose in human. The only thing the EPA knew about this compound at the time was a lethal dose computed in the 70s in rats. And if you feed a rat 825 parts per million food with this stuff for a couple days, you'll kill the rat. And that's it. That's all we knew, that one fact. So the EPA had to quickly respond to this potential disaster and figure out, well, is this safe? And the way that one reasons through toxic compounds in this setting is you say, okay, I know what it does in rat. Now I'm going to apply an interspecies correction factor to sort of figure out what it might do in human. And that factor, there are some justifications for that, but it's more or less guesswork. And that's factor of 10. So you say, okay, so maybe 82.5 parts per million will kill a person. And then you say, but of course not all people have the same susceptibility. Maybe the elderly or infants or infirm are going to be more likely to get sick or die from this than healthy people. So they apply another correction factor, another factor of 10, an intraspecies correction factor. So that gets it down to 8.25 parts per million. And then, of course, it may have effects short of outright killing you. So you put in one more correction factor, which just happened to be 8.25. And they figure, okay, the safe dose is 1 part per million, which just happened to be the level in the Elk River at about that time, so they could stop trucking in bottled water. So that's where our understanding of toxic substances is in the US right now, and Europe and the world. We don't understand a lot. And it really is a fascinating problem because lots and lots of new compounds come to market every year. We've heard a lot about nanoparticles at this conference. There are 500 new nanoparticles that hit the US market every year. And those have actually more stringent regulation than most. 20% of them have to generate toxic data. 80% do not. They come to market without any new data generation. And we can go online, and graphene's great because graphene's really safe. You know, love graphene. But carbon nanotubes, for instance, you can go online right now with your Visa card and buy carbon nanotubes for $250 a kilogram. Google it; it's hilarious. You know, you can just buy the stuff. And this is a chemical that causes necrotic lesions in the lungs. Whether you eat it, drink it, or breathe it, about 40% of what you take in is going to lodge in your lungs and creates necrotic lesions. And we still don't have any legislation over it. And what's more, if you make double-walled carbon nanotubes instead of single-walled, virtually all the same electrochemical properties, same applications in batteries, solar cells, you name it, none of the toxicity, right? And so it's one of those things where a little bit of knowledge can potentially overcome an environmental problem, because it's not like there's anything in nature that knows how to degrade a carbon nanotube. You know, this is a very new compound that our ecologies are having to deal with. And some of these compounds have extremely broad ecological distributions. Pesticides, which I care about a lot, and we're going to talk a lot about pesticides today. Pesticides have enormously broad ecological distributions. Atrazine, for instance, which is used a lot in the US and China, is illegal in Europe and a lot of the rest of the world. But atrazine is used on broadleaf crops, particularly corn in the Midwest. 33 million Americans drink water every day contaminated with atrazine at or near what we recognize as a maximum safe dose. And that's really, that's a lot of people drinking contaminated water.
So that's sort of scary to me. How many industrial compounds are released that we know about, and how many do we really know anything about?
That's a great question. So there, depending on who you ask, there's roughly 100,000 industrial chemicals in use, and we have toxicity testing data for almost 7% of those. So another way of saying that is for 93% of compounds in use, we know absolutely nothing about what they do, usually not even the oral dose for a rat. And even, go ahead.
So we know that Monsanto and Dow, DuPont, Syngenta, and Bayer Crop Science are investigating these things, or just turning a blind eye to it?
No. Those guys, the ag is a little different. So there are 3 sets of laws in the U.S. governing compounds. Industrial compounds are governed under TSCA, which has just been rewritten in the U.S. And then pesticides are governed under different legislation, which is much stricter. So to bring a new pesticide to market, as many of you know, the testing is quite rigorous. But still, it's limited to a few species at a few trophic levels. So like you'll check its effects on an algae, you'll check its effects on something that lives in soil sediment, a microcrustacean in the water, fish, et cetera. And then you rock and roll. You don't necessarily gain an idea of what it's going to do to broader ecologies or how it's going to move through the environment or how stable it's going to be.
So we don't know how it's affecting the ecosystem at all?
That's right. We're not necessarily, which is why we see things like algal blooms, because we don't have a comprehensive understanding of how these things affect the troposphere. So, you know, the U.S. spends $2.2 billion a year cleaning up algal blooms, and that doesn't count any of the other societal costs associated with people being exposed to a neurotoxin.
So we just saw that in Florida and various other places with the runoff going into the ocean and things like that.
Exactly. And so the reason this is so challenging is because toxicity testing is enormously expensive and it's enormously slow. It's also grossly inaccurate, so it's not a good deal. To toxicity testing for one compound takes about 5 years and costs $1.5 million. It's a lot of money. And all you learn at the end of that is, oh, this poisons a rat. Oh, this causes cancer in a mouse. But mice, the cells of a rodent spontaneously immortalizing culture. That is true for tiny critters. It is not true for big, long-lived critters. Humans have 3 primary barriers to oncogenesis. So you can cause cancer in a mouse by looking at it wrong. And the same is not true in primates. It's estimated that 20% of quote-unquote known carcinogens are completely do not cause cancer in higher primates, including humans. And that number is based on 30 years of epidemiological evidence. There's nothing to suggest that they're carcinogenic. So it's not just a problem of underregulation. There's a huge problem with overregulation as well, because the testing paradigms that we have are grossly inaccurate.
So how deep are you digging? I think you got a slide that probably looks at that. So you're digging pretty deep and looking at the effect of these things, aren't you?
That's right. That's right. So essentially, where we are is we're in a paradigm that is retrospective toxicology. You develop a compound, you do limited testing that really isn't very informative because you're mandated to, bring the thing to market, and then maybe 30 years later you find out it was bad or not. And what we have to move to, I propose to you, is a different paradigm that's prospective toxicology, so that we have accurate and cost-effective assessment on timescales that are relevant to industry, because our timescales right now are completely irrelevant. You want a 30-year lag in bringing to market? It's ludicrous. And so we have a strategy for that. So this is a slide courtesy of the Consortium for Environment Care. It's a consortium for which I lead the analysis. It's 29 partner institutions and a couple dozen corporations. It was formed a few years back. And the strategy that we propose is a fundamentally new one for toxicology, and it's the same one we're applying to agriculture. So we're almost back to agriculture. The idea is that instead of doing testing in one or a few species at a few trophic levels, you do testing in a selected cohort of organisms that provides broad coverage across the phylogenetic tree. And this is a very different idea than was done previously, because we can do things like make lots of molecular measurements in many different species being exposed to a compound, impute metabolic networks and gene regulatory networks that are impacted by exposure to this toxicant, associate those with phenotypes in the individual organisms, and then use our knowledge from evolutionary biology. Look at where these networks evolved, where these biochemical networks, where these gene regulatory networks evolved, and project our findings onto the phylogenetic tree. Project our toxicological findings onto the entire tree of life. Now, we cannot make measurements in in even a handful of the ecologically relevant animal species, right? There are many thousands. It's completely impossible. So it's a fundamentally computational problem. We must use a strategy like this to impute if we want to safeguard our ecosystems. For soil and ag, this problem is exponentially worse. We're talking about several thousand species of animals, and more if we get into beetles and insects and things like this. In 1 cubic millimeter of soil, there are more than 1 million species of microbes. And the relationship between the soil microbiome, the plant root microbiome, and agricultural yields is something that we're just starting to appreciate. And it's a really critical relationship that we're going to talk some more about. If we want to keep from unexpected crop failures, algal blooms, and things like this, it's not enough to understand the effects of toxicants on even a few thousand species. We're talking millions. And so it really does call for a phylogenomic approach like this.
So you're talking about a totally new model system?
I'm talking about a totally new model system for understanding the bioactivities of chemicals. And this relates to a general field that was coined in 2008 by Reyes called molecular ecosystems biology, where the idea was that we would take all of the tools from molecular biology, from cell biology, which has been enormously successful at linking the information encoded in genomes to cell function, organ function, animal physiology, things like this, and try to take the next logical step in our understanding of genetic systems and our understanding of life, to go beyond the cell, beyond the tissue, beyond the individual, to the level of the population and the ecology. So how much information encoded in a genome deals with how the animal lives in its environment, or how the plant lives in its environment, or the microbe? And the answer is we think it's a hell of a lot. And so now we're working to understand how gene regulatory systems transduce, respond to, and ultimately influence both populations and ecologies, right? But that's an enormously, enormously challenging problem. It's very challenging because in molecular biology, we have these incredibly powerful tools called model organisms. Model organisms go through predictable developmental trajectories where, they're an egg, then they're fertilized, they're an embryo, they're, a larvae, and then a critter, whatever you like. There's no analog of that in ecology or agriculture. You can build a terrarium, you can build a mesocosm, things like this, but there is not yet a way to take an ecological system that is small and cost-effective and an accurate representation of what happens in the field, and set it up in Berkeley, set it up in London, set it up in Berlin, and have it behave exactly the same way, go through the same state-space trajectory, be fundamentally reproducible. And so that's what we're really trying to do. We're trying to link very complex and realistic systems like mesocosms or acre plots, things like this, to representative terrariums, to things we call ecofabs, which are modified rhizotron systems that allow us to make reproducible molecular measurements in time series. So we can measure the flow of metabolites, the flow of gene regulation, interactions between plants, and the thousands of microbes on which they depend for their subsistence. And then by using both very simple systems like microbial co-culture systems and very realistic systems like mesocosms to go back and forth and try and understand how to build more and more accurate models to yield better and better translation to agriculture. Now, we make lots of different types of measurements. We do proteomics, metabolomics. We look at transcription, of course, and we look at genetics of all of the species involved. In one of the pilot ecofabs, there are a couple. One of the pilot ecofabs run by my colleague at LBNO, John Vogel, we've got Brachypodia, which is a model crop for biofuels, nice tractable grass, good genetics. And 92 microbes. And just 92 microbes are sufficient to reproduce the wild-type behavior of the plant. So it's an extremely tractable system. And what we're working on doing now is obtaining genetic mastery over every one of those 92 microbes so we can fundamentally interrogate the system. And one of the ways that exposure biology comes back into this and toxicology comes back into this is in genetics, you learn about a system by perturbing one gene at a time. But when you're talking about an ecology, that's not enough, because you're interested in the context. I'm interested in a biochemical pathway. The degradation of atrazine by soil microbes is often done by a consortia, no one microbe. That's certainly true in insect guts. We've shown that. It's actually a consortia of microbes. So it's not enough to knock out one gene. You've got to perturb entire systems. And so we use exposure biology or toxicology just like you'd use genetics. We design a chemical perturbation to modify a pathway, and then we study how that changes the metabolism, gene expression, et cetera, of all of the organisms involved in the system. And that's how we learn about new genes. We've discovered 1,000 new genes in Acetobacter tropicalis, for instance. And we're trying now to understand what those do and how they work.
Can you talk about the consortium of environmental care, environment?
Sure.
That you've done? This is so important. I think we need to look at this long-term as a global initiative because this consortium is so important in this study.
Absolutely. So the Consortium for Environmental Care is interested in using systems such as those I've described and using molecular measurements in these systems to try and understand at a molecular level how chemicals and compounds affect our environment. The up behind me is a glyph of the sort of analysis strategy that we use for that, and I won't belabor it. We're a little short on time. But the idea is that you associate networks of genes, potentially in consortia of organisms, with networks of metabolites. Go ahead.
So what I'm getting from this is I think we're in trouble. And can I switch to China for a moment?
Sure.
5 years ago, China spent $53 billion buying land in South Africa to grow food for their people. They've taken over Africa. Everywhere you go in Africa, you see a Chinese complex or something like this. It's to extract not only the goods and services out of China, but also the agricultural products to feed their people. They're only 28% arable land in China. So it seems to be a problem, and can you sort of take this to the other level and say what's happening in China and why.
Sure. So China, as many of you know, has even looser regulation than the U.S. has, and even more inaccurate testing. So in China, about 60% of freshwater rivers are completely unpotable, polluted beyond recognition. 80% of their wells are now unusable for drinking water, 80%. So 550 million people in China have no access to clean drinking water. In China, about 1.6 million people die every year from pollution-associated illness, okay? That's a huge number. That is a huge number. That's much, much higher in the US. That's 17% of all deaths in China. Can you imagine if 1 in 5 of us in this room was going to drop dead from an environmental factor that we could not control? So shown here is a plot from some colleagues of mine at Berkeley that just shows air pollution over a few days in the Beijing area. And the dark red patch in the middle between Beijing and Langfang, that is beyond horrifying. That is an emergency alert, stay inside, you can't go outside, toxic air day, okay? Wow. And where that is located is not in the heart of a city, it's over agricultural farmland. So this is now affecting, beyond a few metropolitan areas. It's a very serious problem for China. China also has a massive problem with algal blooms, much worse than we have in the US or Europe. And so it really is a very critical problem there that we start trying to understand how to mitigate pollution, not just here at home, but in China and more globally. The scale and scope of the problem is absolutely enormous. This is a problem that was created by the Industrial Revolution, and it's going to take another revolution to put the genie back in the bottle, as it were. And the name, we think that sort of serves as a good emblem for that, is environment care, to analogize to the healthcare industry. We want to create an industry around serving the environment. This can't be done by public monies alone. The problem is far too vast and far too global. So how do you do that? Well, people are already doing it. There are people in this room making money off environment care. David, I think you have something that I would classify as environment care.
Yeah, that's right.
Yeah.
I'm not supposed to talk about it.
I see. Okay, then I'll give another example. So John Riles, in his other life in Ag Biome, they have a new compound called Howler, which is a fungicidal microbe that is a substitute in, for instance, organic farming for things like lime sulfur. So organic farming, a lot of folks think, oh, organic, no pesticides. It's not true at all. There's a laundry list of pesticides that are used in organic produce that are approved by the EPA on this list of things you can spray on crops and still call them organic. And essentially what they are ancient. So they're not the best things or the safest things, they're just things that have been around a long time. And lime sulfur melts your bloody skin off, okay? People die from exposure to lime sulfur every year. And companies are switching to completely safe, renewable, biotic compounds. There's another startup in California is competing directly too on the chemical side, so not a microbe, but a chemical out of Merced run by a fellow named Ron Williams. And they've just got a safer form of the sulfurous compound that doesn't melt your skin off. And companies are switching to these compounds, and many of them are substantially more expensive than the traditional chemical alternatives. So you ask, why are they switching? Is it out of the goodwill? No, it's actually not. I don't know about John's business model, but I know about Ron Williams. The insurance rates get lowered. The farmers' insurance rates get sliced when they switch from highly toxic compounds, which have high application fees and high risk associated, to low risk, safe, environmentally friendly, and completely renewable compounds that don't nuke the soil microbiome. When you spray atrazine or even Roundup, Roundup is the safest bloody pesticide, chemical pesticide. You can drink a glass of that stuff, okay? You're okay. It's going to nuke your gut microbiome. Your microbes rely on the same pathways that they kill in plants, but it's not going to kill you. Even Roundup nukes the soil microbiome, and we still don't completely understand the effects of that.
Well, Roundup is a glyphosate, and what happens when you spray glyphosate on a GMO crop, it allows the root system to not take that in and not kill the plant, because corn and beans and things like this are grass, and that's what you want to kill around the corns and beans and things like that. But glyphosate leaks out of the root systems and starts to concentrate over time. So if you have, like, for instance, a grape vineyard and all of a sudden you have a hard freeze and you've been using Roundup or glyphosate, what you'll have is winter kill. And what happens is that glyphosate then concentrates on that root system and kills the entire thing. So now you've got to spend about $12,000 to $15,000 per acre to re-establish that, and you have to wait 5 to 6 years before the grapes are ready. When you can use something that is an organic product that doesn't nuke the soil, but it starts to bring the life back, the enzymes and the microbes in the soil. And that's exactly what Ben is looking at in his studies, to how do we safely put things in the soil that's going to relate to what we eat.
Exactly.
And if you look at what we are putting as poison on our plants, what's going into our system? It's a little bit of it like a poison system. So our biome is fighting a war in so many different directions, and I think that you can relate that to ADD, you can relate it to undiagnosed diseases and things like this that we've been talking about. So this study that Ben is doing is so important to the future. I think that's why Mark and Larry started saying, why do we have agriculture on the agenda? And it's because I am honored, one of the smartest guys I've met in a while, and this is a smart room. But Ben, tell us a little about the environmental consortium, just to tee this up at the end.
Okay, sure. So I'll be quick. So it's as I said, it's an international consortium. It was started out of Europe, but it's got 12 partners in the U.S. now. China has actually come on board with this, and they are extremely interested. They recognize the severity of the problem, and they are starting to embrace these sort of environment care solutions. There's a Danish company selling giant air filters to Beijing, for instance, literally like an air filter you'd put in your living room, but gigantic. But that's, of course, a Band-Aid, and not a fix. Norman Yen in the consortium has a solution in Canada for algal blooms that works very well there. He's got a daphnid, a little microcrustacean you can put in a lake, 20,000 gallons put in a lake, ameliorates an algal bloom in 28 days. So the consortium is very actively working on developing these methods for remediation that can be profitable, can be adopted by industry. Right now we're pursuing the MacArthur 100 and change award. That's a $100 million grant from MacArthur. They're only going to give one. No idea if we'll get it. They probably give it to precision medicine. Everybody does. but if we get shortlisted, it's good advertising. And so the consortium right now is funded by federal monies and a few public-private partnerships. And it's something that's an evolving beast, and I hope you'll be hearing a lot more about it in the coming years.
Thank you. We got time for a couple of questions. I know Larry has one. I can count on Larry.
Ben, this is just extraordinary, and I love the team you've got at LBNL. In fact, I've just been added to the advisory board, so that'll help me help you. But the thing that I wanted to hear your comment on, which David just touched on briefly, is that you're looking at the impact of all these chemicals on the soil microbiome, but when we ingest those through the crops, our human microbiome, it's totally different than looking at human cells, which has been the traditional way, right? Can you talk a little bit about where that's going? Because it sounds to me like the technique you've got would be able to deal with that.
So that's right. So within INSEAD, Bon Thomas in Norway is working on this. We're working on this at LBNL. We use a mouse model and a Drosophila model. Drosophila is very high throughput. Mouse is slightly more accurate for human. And any pesticide that you eat, even in small quantities, impacts the microbiome. So atrazine, for instance, at Midwestern well water levels, remodels the mouse microbiome in a genotype-dependent fashion. And so it's different for different individuals. Some mice are much more susceptible, some are less. It has a substantial effect on mouse neuromuscular coordination at levels that people are eating. So there's actually an epidemiological prediction there. Folks in Iowa, folks in Colorado, parts of it, folks in northern Texas, ought to be slower and clumsier than people in general. And we're working on designing an epidemiological study to see if we can confirm that. What's remarkable is that even though the microbial clades that are killed off by atrazine are different in mice and Drosophila, the phenotype is very similar. We see neuromuscular discoordination in fly. However, it makes the mice sleepy and passive, and it makes the flies hyperactive. And we don't know why that is, but the bottom line is you get a good phenotype that tells you something about the mammalian phenotype in a very efficient screening system. So we're working on developing more screening systems for that. So far, everything we've tested has consequences. We've got phenotypes for paraquat, glyphosate, and atrazine, ethoprophos, and a few other compounds.
We're out of time, but would like to suggest that we look at this connection as a global initiative, looking at the health of agriculture and the health of the human as one symbiotic relationship, so we look at that in the future. Dr. Brown, I thank you so very much. It's such a pleasure. Thank
Really
you.
appreciate it.
You bet.